Over the last century, the nitrogen cycle has been dramatically altered. Nitrogen in synthetic fertilizers, intensive agriculture practices, and fossil fuel emission has seeped into many aquatic environments, where it alters stream functions and dynamics. Despite policies to prevent nitrogen pollution, nitrogen concentrations in stream and groundwater have increased across large areas of the United States and are expected to continue to do so. Much of what we know about how stream water quality changes over time comes from studies where the surrounding land has been altered by human uses. In contrast, we know little about temporal trends in nitrogen dynamics in streams with minimal human impacts. These streams can serve as references for evaluating catchments more heavily modified by humans. The U.S. Forest Service maintains a network of experimental forests and ranges. For decades, researchers from many institutions have been collecting data on the hydrology and stream chemistry from undisturbed watersheds in this network. These watersheds provide unique opportunities to evaluate long-term trends in the relative absence of changes in land use or land cover. The Experimental Forest Network covers biomes throughout the United States and Puerto Rico. Some sites have 40 years of uninterrupted data, and at most sites, stream samples are collected weekly. In this study, we analyzed stream inorganic nitrogen from 22 forested reference catchments in 7 experimental forests. The sites represented diverse climatic, hydrologic, and vegetation conditions, ranging from the tropical conditions of Puerto Rico to the humid but cool Pacific Northwest. We found that stream nitrate declined in the Pacific Northwest, in the Northeast, and in Puerto Rico. However, these trends were not consistent at national or at local scales. Some sites in the Mountain West and the South showed increasing trends in nitrate concentrations. Adding to these complex spatial patterns of trends, catchments within an experimental forest sometimes had opposing trends for the same nitrogen species for the same period of time. This suggests that the controls on stream nitrogen concentrations may vary among and within experimental forests, and that extrapolation of trends from one catchment to a neighboring one should be made with extreme prudence. We also found that the inclusion of additional years in the analysis changed the direction of the trend, and that stream nitrate and ammonium did not show consistent trends or coincident timing in trend shifts. This highlights that even when trends for short periods are statistically significant, they are not necessarily indicative of longer-term patterns. Caution is needed when extrapolating trends over time and across species of nitrogen. Long-term research and reference sites provides a unique opportunity to document changes in nitrogen concentrations. Understanding how nutrient concentrations are changing over time in reference streams is vital for informing best management practices that are aimed at protecting water resources.